want to say I appreciate it, and thank you all for the very nice honor. Ron mentioned that humans are in a competitive environment with other species. I thought I'd mention one of my favorite illustrations of competition. It was about a retiring samurai in Japan who was the emperor's #1 samurai. So the emperor decided to have a contest over all of Asia to find his new samurai. And they had local contests, and the finalists came to appear before him. One was from China, one was from Japan, and the other one was from Israel, of all places. Israeli was Matsumoto Goldstein. And he went last, but the first from China came up. He appeared before the emperor, and he took a little box from his vest and let out a fly, took his samurai sword, swished through the air. The fly fell dead to the ground, cut exactly in half. The crowd oohed. And second was the Japanese samurai. He came out, took a little fly from his box, let it loose, and swish, swish twice, and the fly fell to the ground dead, cut in exact quarters. And of course, the crowd went wild then, and the emperor thought that must be the winner. But then Matsumoto Goldstein came up. He took out his little box and fly, let it loose, took his sword, a swish, a mighty swish through the crowd. Ripples of air went through the crowd, but the fly continued to fly. And the emperor said, "Matsumoto, what kind of a samurai are you? The fly's not even dead." He said, "Dead? Dead is easy, but circumcision." So that's my lesson to the young people here today. Set your sights high. Okay. So we're going to talk a little bit about co-activators, and these are the molecules that make the receptors Ron talk about work. And although some years back when they were discovered, and the first one was cloned in our lab, they were considered maybe a biochemical complication to learn. But the numbers and the relationships of these molecules to physiology and disease is enormous. And they're having great relevance to medicine, and also will be a new class of drug targets for the future. So I'm going to pick out one today, SOC3, and tell you a little bit about that to have a focused story. So this is just, for those of you not familiar, a general picture of the co-activators. They're large proteins. They have on the N-terminus a protein-protein interaction domain where they work on a large complex of proteins, and basically most of them are enzymes. There's almost 500 of these co-activators and co-repressors now, which we as a group call co-regulators, and they form these large complexes and they regulate most of transcription, but other reactions in the cell too. Why SOC3? Because SOC3 is the second most overexpressed oncogene among all oncogenes now, second only to c-Myc. This is a little example of the overexpression rates in just these cancers here, with over 30% amplification in breast and ovary, and over 70% amplification of the gene in colon cancer. Enormous impact in cancers. The molecules work. I'll tell you a little bit how they work. I'm going to actually show you a picture of some of them now that we can look at. And then we will also talk about a little bit about maybe how we might regulate these molecules in the future. So SOC3 is a molecule that can go in and out of the cytoplasm, and it's subject to activation and regulation by many different pathways. I show three here: estrogen, inflammatory pathway, or growth factor pathways like HER2 or EGF receptor. Each of these regulators has downstream kinases, and when they hit this molecule, they introduce different phosphorylation patterns in the molecule. This introduces a code that sends it to different genes, and with different complexes on the genes. So you have coordinate regulation of many, many genes for growth turned on at the same time. Okay, so that's the transcriptional response there. But SOC3 also has all the characteristics for a growth regulator. It also regulates cell motility and invasion, the other potential things you need for a cancer cell or for a normal cell to grow. So I'm going to show you just by quick illustrations without going into a lot of data what we found out about how this, in fact, works. The SOC3 works for motility out at the membrane, not in the nucleus, not driving just transcription factors. In fact, there is a spliced form of it here, missing the nuclear localization signal, that comes from here out into the cytoplasm, couples up with a kinase associated with EGF receptor, and is phosphorylated in two places. Then the N-terminal phosphorylation interacts with EGF receptor. The C-terminal interaction with FAC, the main focal adhesion kinase for motility. And then you integrate with integrins and you have cancer cell migration and metastasis. Who would have thought you have a transcription factor actually working in a membrane in a coordinated fashion? So it's turning on the growth genes and also turning on motility, but now a piece of that protein conducted by the gene is going out to the membrane. Okay, but it also has ability to direct invasion of cells too, which is another process required for normal growth and for cancer. And this occurs through interaction with a kinase called ERK3 kinase. We found this proteomically when we isolated SOC3 and found that it was bound to a kinase ERK3. I thought, well, we must know a lot about ERK3 because ERK1 and 2 are the MAP kinases and everybody studies those. Well, ERK3 was an orphan kinase. Nobody knew what regulated it, nobody had a substrate for it. Well, what happens is it actually phosphorylates SOC3 at one site, serine 57. This now causes it to go and bind to PEA3 transcription factor in the nucleus selectively with that 857 phosphorylation. PEA3 turns on the MMP genes, the metalloproteinase genes used to digest the membrane for invasion. So again, now you have this same gene directing multiple forces. And that's why we tend to think of these as master genes, genes that are able to regulate many different subfunctions in the cell and in different compartments of the cell towards a given physiologic goal or function. For instance, now, if you look at this, then ERK3 and SOC3 collaborate for lung cancer invasiveness. So if you look at an aggressive invasive model, you see many invasions in the animal in the lung. If you knock out or knock down with shRNA, ERK3, you see greatly reduced invasions. If you knock down SOC3, greatly reduced invasions. If you knock down them both, you have virtually no metastasis here. So it is a potent agent for metastasis. I'm not going into all the details how it does this, but it's the collaboration of this phosphorylating this that causes this when it's not there. Can there even be more to SOC3? Yes, there can be. Chemoresistance, of course, that is a big thing in clinical therapy. Here's one example of this in human beings. This is ovarian cancer patients. These in blue are patients that are sensitive to the therapeutic agent carboplatin, and in green are the patients that were resistant to it. And then these are the levels of SOC3 in the cell, and you see all the sensitive patients had low levels, and most of the resistant patients had high levels of SOC3. And of course, it's known to make cells resistant. So we wondered, what is this? How is this occurring? Well, what we did was take memory cells and we knocked out SOC3 in the cells. Okay? They now are very sensitive to chemotherapeutic agents, where with SOC3 in them, they're much more resistant to chemotherapeutic agents. So we now have created a sensitive cell minus SOC3. To find out what SOC3 might have been doing downstream in the cells to make them resistant, what we did is transfected a whole entire genomic cDNA library and looked for colonies that now survive with the drug administration that now could resist chemotherapy. And what we find when we do that, the bottom line is the major protein downstream is a protein called TRAF4, which initially we didn't know much about. It's a distant variant of a molecule that mediates TNF responses in the cell, but it's not involved with TNF. So what does TRAF4 do? Now, we had some fun with that. Well, we went through a lot of data. This is recently published, so I'm not going to show you the data here, but just the summary. It's an intricate but a very exciting pathway that occurs. Okay, normally in the cell, you have P53 is one of your guardians of the cell, of course, and it regulates apoptosis in the cell. So if the cell doesn't want apoptosis, they mutate or get rid of SOC or P53, as you know here. And of course, one of the regulators of P53 is MDM2. When you have cytotoxic stress in the cell, MDM2 is a ubiquitinase of P53. It ubiquitinates P53. That, of course, causes the cell to get rid of P53. But there's a protein that antagonizes this response in the cell called HOSP. HOSP is a deubiquitinase. So it can antagonize this response and get rid of the P53 ubiquitination, thereby maintaining P53 levels in the cell. So it's working against the cancer process, huh? So what you have then is apoptosis going on rather than substantial cancer development. However, when you now have SOC3 at a transcriptional response induces the gene TRAF4. It uses AP1 transcription factor, but we don't have to go into that. Anyway, so it produces TRAF4 in the cell. TRAF4 is a strong competitive binder of HOSP. So it competes with HOSP, pulling it away from P53, and thereby allowing ubiquitination to occur, blocking apoptosis, and then what you have is resistance to chemotherapy. So this is an amazing number of things occurring all due to one gene, one very strong anabolic and oncogenic protein occur. Again, evidence for multifactorial oncogene and master gene. Okay, just to summarize some of the things again quickly before I go on to show you something else. So you have upstream activators for SOC3. And of course, I showed you one of the things it could do is regulate transcription and turning on growth genes. With data I didn't show you, it also can affect the correct splicing with other proteins on the gene of those genes, target genes. So you get more and you get appropriate splicing and message to produce the proteins. And that, again, I didn't go into. It can also regulate message translation, favoring certain kinds of messages for physiologic response. And that's, of course, published. It also, and other things I didn't mention, generates new mitochondria in the cell. It also causes vessel production outside the cell. And as I showed you, it affects motility of the cell, invasiveness, and chemoresistance. Look at this panoplia of effects coming from this one gene, and now you can see why we call it a real master oncogene and why so many cancers want a lot more of it, because you don't have to mutate it. You don't have to worry about all these other responses. Just make a lot of SOC3 and saturate the response. Okay, now I'll show you something that's unpublished, but some of the first times I've shown you this. Would you like to see what these things actually look like? We're always drawing them schematically, biochemically, but using cryo-EM now in studies, we're able to, with the purified proteins, reconstitute the primary complex and look at it. So I'll show you the structure of an estrogen receptor, SOC3 and P300, another co-activator, all in a complex on DNA. This is what it looks like. This is the ER dimer, the green here, facing this way to you. There's 2 molecules of SOC3 shown in red. So 2 molecules bind 1 ER dimer. And on top, 1 giant P300. Of course, that's the acetylase brought into the reaction here too. That's, if you turn it now just another quarter around, you see this comes out to the front here, and you can see the other side. Now you can see the 2 dimers here of the estrogen receptor. And those are just the pictures of the way they come out before in cryo-EM they go through high resolution calculation. Okay, how about show you just to finish a little dynamic movie of all of this here, in which we look at this structure. Where's the movie? The movie's supposed to come on. Could you turn it on up there? No? Well, it worked in the project. Oh, here it is. Okay, there you go. There you go. That's the complex as it comes out. And look, the resolution shows there the blue's P300, the 2 SOC3s, different color, and ER dimer. So as you can see, they have their own structures. This is the structure they have in the complex, and you can see how they fit together there. Okay, you can see the 2 SOC3s, of course, are similar, but in the complex, they do not touch each other. And there's 2 of them, not 1, as some people have said, 2, but they're not. It's not a dimer, it's 2 independent SOCs and 1 P300. This now, if you add in the DNA and some of the structure of the ligand binding domain of ER, you see this there, and you see the SOC coming in. It binds directly where it's supposed to on the receptor. At AF2, it's interacting with AF2 here in the molecule. And then coming in on top, you'll see, again, would be P300. So, of course, this is occurring very fast in the cell, and that's a rather dynamic picture. So this will never be probably seen by crystallography in your lifetime because you can't do this with crystallography, but it can be done with special care in a procedure we worked out over the last few years with cryo-EM with Hua Chu, our excellent cryo-EM person at Baylor. Okay, so just as a quick summary, here we go. This is the estrogen receptor sitting here with the ligand binding domains in AF1 and the DNA binding domains on there. 1 SOC3 comes in, binds to 1 LBD or AF2. Another one comes in, binds to the second LBD AF2 on the other side. They do not touch each other. Then coming on top is P300. And you see, you could mix and match these because they're not a dimer. You can have, which we have found on genes, sometimes it appears that SOC3 and SOC1 there, well, they can both fit, and they can go to the different sites. Or you can bring in other co-activators. Carbon-1 also will come in as a dimer at that site in studies we're doing now. So, and of course, this is a basic complex that then many more proteins come in on top of this, and you have a giant transcriptional regulatory complex at the enhancer. So it's It's exciting now to be able to see this instead of we just draw it and suppose it from our biochemistry experiments. And there's a lot more things we've learned about this that I don't have time to go into. So finally, to finish up just a bit, now that we know this is a bad actor, ACTR, as Evans would call it, can you do something about it? Could we possibly regulate it? Well, many people have said no, that this can't be really done, that these are large molecules, they don't normally bind ligands, and who knows if you could bind anything to regulate them. Well, since this is such a major oncogene driving growth in the cell, what we would like to do is get rid of it. So could we find molecules that actually bind to it and cause it to be degraded selectively in the cell? And the answer is yes, and we published just recently a couple papers, and I have a couple more of these coming, in which we find better and better molecules to regulate this process. What's the theory here? Here's the theory. It's pretty simple. Let's take a breast cancer here. There are many, many, many different downstream transcription factors regulating genes to produce growth in a breast and whatever else the breast needs to survive. Sitting above them and activating all of them is SOC3. When you interrupt this pathway like ER with, say, SERMs or aromatase, you could get a response, you get regression, and then other pathways are free to upregulate and the cell comes back and you get resistance. Same thing when you treat with Herceptin or so forth, this pathway, the other pathway that's still left to compensate and upregulate. The idea is that if you can hit this, you'll hit them all simultaneously, and maybe you'll compromise the cell to such a degree that it can't survive. And in fact, that's what we found when we knock it down with SOC3. If you take a cancer cell that overexpresses and is addictive to SOC3, and you knock it down acutely, it'll kill itself because it has no easy escape pathway, no out. So I'll show you a couple compounds. I'll show you some names here in a minute, but this is one of the first compounds we found here, that what you can give in a cell with here and 10 micromolar concentration, you can knock down SOC3 in the cell. It also knocks down SOC1. That's actually good because SOC1 is an oncogene too. We'd like to get rid of both at the same time. However, you don't get an effect on P300. The other co-activators like CARM1 acted in the cell and so forth, and not quite as much on SOC2 at all. So these are all, that's the type of thing we're after. And these molecules were shown to bind directly to SOC3 and cause its degradation in the cell enzymatically. If you give it to various cancer cells and you look at cell viability, not growth viability, you see it kills most of the cells in increasing doses, okay? So we're after a death response in cells that overexpress SOC3. Here's another one, liver tumor cells, same kind of a kill curve. When you add the compound in, but maybe it's just killing everything and super toxic. Well, these are primary hepatocytes now. They don't overexpress SOC3, and you see no effect on those. Okay, and we found this repeatedly depending on the concentrations in the different drugs we find. We, of course, have gone on to show other drugs. We went to the Chinese literature and we found a component in Hao Chan Su that actually at quite a low concentration will kill cancer cells. These are MCF7 cells. It's a 3.2 nanomolar IC50 here. And when you give this kind of a compound, which is bufalin, is actually the active ingredient in Hao Chan Su. This comes from a poison toad in China. You see, it doesn't affect body weight or growth of the animals much, but it does greatly diminish lung metastasis in animals and a very aggressive MDA-MB231 subline in xenograft models in the animals. So we have a story here now where you have molecules that we didn't know existed at all a while ago. We now know a lot about how they work. We know the physiology, we know how they're used pathologically, and now we're entering a phase where we're trying to use these as new targets for medicine. And to finish, I'll just say that I'm greatly indebted to the collaborators on the left, are the people that actually carried out different parts of these experiments: David Leonard, Wai-Wen Wong, Ping Yi, Xiao Wang, Anna Malabanaya, Li Wang, and Frank Mado. And also on the right, more senior collaborators, Hua Chu in the cryo-EM, Sophia Tsai and Ming Tsai in some transcription studies, Jae-Ming Xu in cancer, and Pat Griffith for discovery of some molecules. And I thank you again for your attention and for the honor of the Dale Medal for this year. Thank you.
Ladies and gentlemen, the 2014 Dale Medal to Dr. Bert O'Malley. Thank you very much.
Thank you.